a pleasure to welcome Professor Hosoi to Monash University. She is here on a Shelby Fellowship, which is granted by the Australian Academy of Science. Professor Hosoi is the Neil and Jane Pappalardo Professor of Mechanical Engineering, also Professor of Mathematics, and a core faculty member of the Institute for Data Systems and Society at MIT. She's previously been the Associate Dean of the MIT School of Engineering and also had a large number of awards for her work, including from the American Physical Society, the SIAM Block Community, and the Jacob P. Den Hartog Distinguished Educator Award. She's also a Fellow of the American Physical Society, a Radcliffe Institute Fellow, and a McVicker Faculty Fellow. So thank you very much for coming to give our colloquium today, Peko, and I'll let you take it away.
Thank you so much. Thank you guys for having me. I'm having a terrific visit to Australia, so I'm happy to come back anytime. So today I'm going to talk about filtration, and hopefully, with given time at the end, I'm also going to talk a little bit about engineering education. So I'm going to try to save some time for that. Um, Please interrupt me at any point if you have questions. I will watch the time, so I promise I won't go over even if we get into a discussion in the middle of the talk. Okay, so filtration. I'm just going to put up one picture here, and this is just to remind you that how important filtration is. So the first picture up there, that's the Deer Island Water Treatment Plant, which is where Boston gets its water. Those of you who spent time in Boston, that's where your water came from. The other one is just sort of a standard commercial HVAC system. So we spend a lot of effort moving around water and moving around air, and in all of these systems, there's filters. So the more efficient you can make your filters, the more efficient you can make these systems. And one of the things that my group tends to do when we see challenges like this is we tend to take an approach that's sometimes known as bio-inspired design. And the idea behind this is natural systems have had millions of years to evolve. So if they can perform a task that is similar to one that we as engineers might want to perform, it can be instructive to look at what mechanisms they use to do that and see if we can replicate them. So we're going to think about that in the context of filters. So the first thing you do is you look for systems that filter, and good news, there are a lot. There are many, many systems in nature that filter. There is, so I'm going to, let's see, oh, I've got a pointer. So this plot, this is a very complicated plot, but here's the message I want you to take from this plot. There is a dimensionless number on this axis. For those of you who are fluid mechanics and mechanicians, it's a Peckley number. There's a dimensionless number on this axis. It's called an interception number. But at some level, it doesn't matter which numbers I put on here. What I want you to notice is that each of these kind of black ovals is an animal. So that's the range, the parameter range where the animals operate. And each of the red ovals is an industrial system. And so the message to take away from this is that the animals are already operating in regimes where the industrial systems are operating, which is great news, right? So that means that we're already we're in a parameter space where if they're doing something interesting, we might be able to move it over. Okay, that's the good news. There's also bad news. The bad news is that almost all of these animals do the same thing, and this is what they do. That's it. It's very simple. And this is pretty much what we do in all of our kind of most of the large industrial filters is you just have some kind of a mesh, and the small things go through and the big things do not. And that's it. And the problem with that, it works pretty well, is but the problem is that the big things get stuck on one side and you get something called fouling. So if anybody's changed an air filter, your air filter might look like this. And at some point, you have to either clean the filter or change the filter or do something to get rid of all of the stuff that's stuck on the other side. So fouling is a big problem in all of these industrial scenarios. Okay, so I did say almost all of the animals do this, and there is one exception. Well, you guys can come sit up front. There's lots of chairs. Go ahead, jump on in.
Did you say we had to interrupt with questions?
Yes, interrupt at any time.
Do biological systems have fouling?
Yes, yes. So biological systems have fouling, and a lot of times, so they have different like mechanisms for cleaning it off. But mostly, biological systems also tend to have little arms, and so they tend to just do this to clean their filters. Yeah. So yes, yes. But and there's other mechanisms as well, but that's pretty common. Yeah. Great. Yeah. Yes. And thank you. Please do ask questions as we go. Okay, so almost all animals. There's one exception, and that is the manta ray. So here's the manta ray. So this is a video from the BBC. So manta rays, they're filter feeders. And they are, I don't know, you can probably see there's little specks here. So they're sweeping up all of these little food particles. And in a minute, you're going to see the feeding behavior. So this is what they do. They do a big sort of a circle. They do a big arc that comes around here. The food particles go in the mouth, and you can see there's some sort of dark regions, and then clear fluid comes out the gills. The food goes down into the stomach, and clear fluid gets swept out this way. Okay, so that's the system. Here's another one that's coming by. Let me tell you a little bit more about the structure of manta rays. So a manta ray, if you look inside a manta ray, you've got these sort of white things. This is cartilage. So that's the structural supports that's holding this thing open. And between these white sort of structural supports, there's this dark region. And the dark region is the filter, right? So this is where the particles are going back down into the stomach. And the clear fluid is coming out through these dark regions. And if you zoom in on one of these dark regions, what they look like is this. And there's been fantastic work done by Misty Pek-Tran's group. I don't know her, but I've read all her papers and they're amazing. So she has looked at all of the structures in these manta rays. And if you look again in these dark regions, it looks like this when you zoom in. There are these tiny little rows of plates that are just stacked up on top of each other. You can see there's some sketches over here. And the scale of this, you can see that's about 5 millimeters. So these are kind of on the scale of millimeters. These are plates from a bunch of different species of manta rays. And regardless of the species, the plate spacing is about the same size. Okay? And this is actually kind of surprising because the manta rays change size dramatically. So has anybody ever swum with manta rays before? Okay, yeah, there you go, manta ray swimmer. Yeah, they're huge. So they can, like you can see, they can get up to 7 meters. 7 meters is, I mean, think about how big this room is. 7 meters is huge. And then there are smaller ones that are maybe a meter, and then there's juveniles. So these they span sort of an order of magnitude in size, but these are always the same size. Okay, so the other thing that's come out of the biological literature is the following hypothesis. This is how they're using these things for filtration. So this is a sketch of what these look like from the side. So I'm kind of looking at one of these things from the side, and each one of these is a little stacked plate. And the mouth is over here, and the water is coming in this way, and you have little particles. And they have proposed a mechanism that they call ricochet separation, where the little particles ricochet off of these little plates. So they just basically bounce down the panel, then into the stomach. Okay, that's the proposal. I don't know if that's correct or not. But in the next few slides, we're going to develop a model for manta rays, and we're going to see whether this is a feasible mechanism for filtration and see whether or not this tells us something about the design parameters and why these might turn out to be the size that they are. Okay. All right, so I've now given you all the background I know about manta rays from the biological literature. So now we're moving to fluid mechanics. All right, so here's the manta model. Okay, so our manta model, this is our manta ray. I was brought up as a physicist, so my manta ray is a cylinder. And yeah, so it's a cylinder, and the cylinder takes in fluid on this end. And it's going to be a leaky cylinder, so fluid is going to leak out the sides. And what's going to leak out the sides, this is going to be the clear filtered fluid that comes out the side of your cylinder. And the particles are going to go back to the back of the cylinder. Okay. All right, so now we're going to do a little bit of modeling. By the way, how many people here are fluid mechanics people in this room? How many people would classify yourself? Okay, good. How many people are have taken a fluid mechanics class before? Okay, better. Okay, good. All right. So we are. I'm going to do a calculation, which you could put on an exam in a first year fluids mechanics class. So those of you who teach fluid mechanics, take note. You can put this on your exam. Those of you who are going to take fluid mechanics, take note. This could be on your exam. Those of you who have not done fluid mechanics, you're about to learn some fluid mechanics. All right. So there's two questions that we basically want to talk about when we're talking about filters. The first is the permeability, and the permeability is basically a question about how much does our cylinder leak. Okay, and in general, you think about how much power is your pump putting out versus how much flux are you getting through your filter. In this case, the pump is the fact that the manta ray is swimming, right? So it's swimming. So that's the thing that's pumping fluid through the system, and then something is leaking out the sides. So what I really want is I want a relationship between my swimming velocity and the volume flux of the leakage out the gills of the manta. Okay. Okay, so here's how we're going to do it. So I have a leaky pipe. This is my leaky cylinder. Actually, I'm gonna just to keep the algebra as simple as possible. I'm going to do this as 2 plates. But you can do the same thing with a pipe. You'll just carry around a bunch of pies and things. Okay, so I've got 2 plates. I have fluid coming in on this side. I have some Q leak coming off the sides. I have some fluid parameters here. I have the size of my cylinder. And I have something that I'm going to call the permeability. For now, I'm just going to call this K. And the permeability basically says how hard is it for me to push water from this side to this side of my pipe. And we're going to relate this in a little bit to the geometry of those little plates. But for now, let's just say there's some permeability. Okay, so the first thing you're going to do is you have to conserve mass. And so to conserve mass, I'm going to draw a little control volume. So here's my control volume. And I'm just going to keep track of what's coming in and what's coming out. So I have some flux coming in and I have some flux coming out the other side, and I have some leakage out the sides. And if you do some algebra and take the limit, the delta x goes to something very small, you get this equation for mass conservation. So my Q leakage, that's my flux leakage, is equal to H, half the height of the cylinder, times dq dx, which is how much your flux is changing down the pipe. Okay. The second thing we're going to do is we're going to relate the leakage flux to the pressure inside this little box. Okay, so there's some pressure inside this box. And for those of you who have taken a fluid class, you may recall there's something called Darcy's Law. Does Darcy's Law sound familiar to anybody? No? Okay. Okay, some people maybe. So Darcy's Law just says that the amount of stuff coming out is proportional to the pressure drop across this thing. So I push harder and more stuff comes out. Okay, so there's my Q leakage is proportional to the pressure drop. And the constant proportionality here is basically the permeability. So remember, the permeability tells me how hard do I have to push to get fluid from one side to the other. Okay, great. So now I have 2 equations. This one is equation for QL. This is an equation for QL. So I can just set that equal to that. And if I do that, I have this relationship. So I have a dq dx is proportional to P. And both of these are unknown, right? I don't know what Q is and I don't know what P is. So I need one more equation. And so there's one thing I haven't used yet. So I've used conservation of mass. I've used something about the permeability. What I have not done yet is conservation of momentum. So I need to put in conservation of momentum. So I do that. And so for conservation of momentum, there are lots of different kinds of scenarios you can imagine because it's basically flow down a channel. And in that flow down a channel, you can imagine turbulent flow or laminar flow or different kinds of flow. I'm going to do the easiest one again, just to keep the algebra simple, but you can do whichever one you want. So this is laminar flow. So for laminar flow, my velocity, the shape of my velocity profile going down that pipe is parabolic. Profile. I'm now going to just depth average this to get a flux. So I'm going to integrate that, which gives me a Q, and that gives me my last equation. Okay, that was a crash course on fluid mechanics right there. And again, you can see how you could really put this on an exam. All right, so now I've got these 2 equations, and now what I can do is I can just take, for example, this Q and plug it in over there. So I plug this in over here, and this becomes the second derivative of P is equal to P, which is a differential equation I can just solve. Okay, so you solve it. There it is. That's the solution. Fine, it's an interesting solution. And the last piece I need was the thing that I highlighted, which is that I sort of put this K, this permeability, in as a placeholder, saying, okay, there's some parameter that tells me how hard do I have to push on my fluid to get it across this membrane. But that's going to depend on the geometry of my little plates, right? So now I want to relate K to the geometry of my little plates. Okay, I have one more slide of math, and then I'm gonna throw my microphone off my face, and then I'm going to show you some results. So the last slide of math is here. Here are my plates. This is a side view of the plates. They're spaced by some amount d and some period t at the point L. So that's the geometry of those little plates in the manta. Okay. Remember what I told you before is that Darcy's law tells me that my leakage is proportional to my pressure. That's what we had on the previous slide. The constant proportionality is this permeability. I can also say that, well, the way this thing is leaking is I've got just tiny little flows through these channels. And so I can say in each of these channels, I just have laminar flow. So I just have a little Hosoi profile, so a little parabolic profile. And I can just integrate all those little parabolic profiles and add them up. And that will also give me a leakage. Okay. So I do that, I get this expression. And I just have to set, so this is also equal to Q leakage. And so I just have to set this equal to that. So I do that and then solve for K and I get the following. And for those of you who have done low Reynolds number flows, this might look kind of familiar because you'll see that the permeability scales like d cubed, which is the spacing between these little plates. So anytime you have something that is sort of viscously dominated or low Reynolds number flow, this d cubed is a very common scale. Okay, so now I have a prediction. So I'm just going to plug this K into my, this is what I just derived on the previous slide. I'm going to plug my K into that. I'm going to normalize by my flow down the channel and do some algebra. So this is just algebra. And what comes out is, oh, and I'm going to remind you that Q channel is basically the swimming velocity of the ray. And I come out with this. Okay, I now have a prediction. This is my prediction. So the prediction is that the leakage velocity normalized by the channel velocity, which is basically how fast I'm swimming, scales like is equal to this. This term is basically whichever pore number you're on. So X is how far down the manta you are. L is the total length of the manta. And T is the periodicity. So you can convince yourself that I put those together. That just tells you which pore am I sitting at. Okay. And it should scale like this. And so we can check this. So we check it. So, and the first way we're going to check it, I'm going to do experiments in a little bit, but first I'm going to check it by doing a full numerical simulation of this. Do a full numerical simulation of the channel, which looks like this. So I put in some channel geometry that looks like this. Well, me meaning my graduate student puts in this geometry. And I'm going to zoom in over here, and he does these beautiful simulations where the little plates, and this is the color is the pressure field. These are streamlines. This is the little pore that the fluid is coming out. That's the channel. And you do a whole bunch of these, and then just check that scaling that I had on the previous slide, and ta-da! It's great. It works. It's exactly right. So it's bang on. So this line is this equation which we just derived. These are the points from the full numerical simulation. And again, you're just measuring how much is leaking out the pore versus the channel wall. And it works beautifully until it does not work beautifully at all. So the question is, so we understand this piece over here. So, and let me note this piece, this is sort of what happens when either my pores are very tiny, remember that's the spacing over there, or my channel velocity is very low, right? So if I'm swimming slowly or my pores are super tiny, then I get this beautiful scale. Okay. If I start swimming too fast or my pores get too big, then things go awry. So what is happening when you get into that parameter regime? So I'm going to show you what the streamlines look like in the simulation. Okay, so so this is the numerical simulation. This is a simulation where everything is working well. So this is where the scaling does exactly what you'd expect it to do. Okay, this is what happens when it is not working. Okay, so you see that one thing that happens is you get this big vortex which sits at the top of this pore and blocks the flow into the pore. Okay, so why do you get this vortex? Well, so you can kind of see a hint of that in the pressure contours, right? So here you can see my pressure contours are kind of aligned like this. So the pressure gradient is pushing stuff that way, right? So my pressure gradient is going to do something like this. Over here, you can see if I look over here, my pressure contours are kind of going this way. So I'm going to get something that pushes my flow in that direction, right? So I have something that looks like this. And so did my pressure contour switch? Well, you can see right over here, there's a stagnation point, right? So this is something where I've got a streamline that's coming like this. So I have a stagnation point here that's building up a high pressure because this thing sort of slams into this little pore. And that high pressure is pushing stuff backwards. Okay. So whether or not I get this vortex depends on the relationship between the pressure in the pore, the pressure at the stagnation point, and the pressure in the channel. Good. Okay, so the last piece I need then is I need to understand what is the pressure at this stagnation point. Okay, so I have one more slide of fluid mechanics math, and then again, we'll put it all together. So here's the next slide of fluid mechanics math. This is another thing that you could do in a first-year fluid mechanics class. How many people who've taken fluid mechanics have seen similarity solutions in their fluid mechanics class? Good. Yes. Yes. Good. Okay. A few shy people who maybe have done it but don't want to put their hand. Okay. So So this is a similarity solution. The similarity solutions are beautiful. So this is one that was actually done by Siemens in 1911. Okay. And this is for the following scenario. So this is a stagnation point. Imagine I have a stream. This is like a big bath of fluid. I have a stream coming down here. It hits the wall. This thing turns the channel, turns the corner. And sort of far away from the wall, I get a nice sort of hyperbolic flow profile. But right at the wall, I have to connect this to whatever is happening at the wall, which has a no slip condition. Okay, so I need something that will connect this nice hyperbolic solution to the no slip condition. So Siemens propose the following, that in the far field you have this, so that's my hyperbolic solution. And in the near field, he said, okay, I'm going to guess this. This is what I think it's going to look like. I'm going to guess that this is my my similarity variable. So I'm going to do a change of variables to this, where y is scaled like this. And I'm going to scale my velocities the following way, in this particular way. And when you do this, so you do this, you plug this into the Navier-Stokes equation, which is conservation of momentum. You turn the crank, you just do a bunch of algebra, grind through it, and what comes out is a relationship between the pressure, the other variables of the problem. So you get a solution for the pressure, and it only depends on F. And F is the solution to this ordinary differential equation. Okay. And this ordinary differential equation, you only have to solve it once, and then you're done. Right. So once you know the solution of this, you know everything about the pressure. Once you know everything about the pressure, you can go back and you can get the velocity. So you solve it once, and this is the solution. I actually didn't solve it. I think I got this from Wikipedia, because really somebody has to solve it once, and then you have the answer. All right. So so this is what F looks like. So you plug F back in, and that gives you your pressure profiles. Okay. So now how do I translate this back to the manta ray? So the way I translate this back to the manta ray. So remember, this here. So this is a pore, and I've got this big vortex sitting at the mouth of my pore. Okay. The fluid flow. So if the fluid comes in over here, it just gets recirculated over here. So the leaky fluid has to go around this vortex and come out this way, right? So anything that's leaking is coming on that trajectory, which means that my Q leakage, in order to get how much flux is leaking, I have to integrate my velocity over this little chunk over here. Okay, I integrate over that little chunk over there, and that tells me how much gets by. So that's what this is. So Q leak is, I'm just going to integrate my velocity over that little chunk. And now I'm going to just rescale according to Jimenez. So my U becomes this, and these are just, that's K, it's just constant, which gets pulled out. And I have this square root in front of that. And so when I do that, you find that Q scales like the square root of K. Okay, square root of K, what is K? Well, K is something that tells you the scale of the velocity in the far field. Okay, so that tells you that Q is scaling like the square root of K in the far field. What is the far field in this picture? Well, the far field is the middle of the manta. It's the channel, right? It's wherever that fluid is coming through the manta. So that tells you that Q should scale like the channel velocity to the 1/2. Okay, so I've got that. Okay, so good. So I now have a prediction for that region where I have those nice little vortices. And there's something very bizarre about this prediction that I've just showed. So you will notice that in this prediction, there's nothing about the geometry of the pores. There's no D width of the pores. There's no L width of the pores. There's nothing that's going to tell me that the manta should pick a particular geometry. It says that once I get into this region, once my flow is going fast enough, the geometry of the pores becomes irrelevant, which is weird. So we should check it. Okay, so we're going to check that. So this is our bizarre prediction. So here's what this, let's summarize what we've got so far. So remember, for permeability, I want to relate my channel velocity, which is my swimming velocity, to my pore velocity. Okay, for small channel velocities and small pore velocities, I have that relationship that works beautifully, right? So that's over here where I've got this nice relationship where my leakage is just proportional to the channel velocity. Okay. And now I've said, well, if I go too fast, I'm now in a regime where I have a vortex, and in the vortex, I've now claimed, so my leakage is actually going to go like the channel velocity, like this. So now if I do a bunch of simulations or I do a bunch of experiments, each one of those is going to be a plot on this, a point on this plot. In this region, I should see this, by the way, this is a log scale. So in this region, this is linear. So I should see a bunch of things with a slope of 1. as I change channel velocity, but hold the geometry constant, right? So this is for one D, this is for another D, this is for another D, this is for another D. But as I move into this region, everything should collapse on the one line with a slope of 1/2. Okay, so that's what we expect. Okay, so if this is correct, so we do that, and here's what you find. Amazing! It does exactly that, right? So it is true that once you get up here, the geometry of the pore doesn't matter. It doesn't care. That doesn't care D. It doesn't care the plate size. Down here, the geometry matters, and over here, something complicated happens. There's some transition where you have to connect. Okay. So we think we understand that pretty well. Of course, you have to check this in experiments. So we did the experiments. So this is our mini manta. So this is the manta that sits on the desk. There's a flow that comes through the middle here. It's about this big. These are the pores on either side. And you collect whatever's coming out in these little tubes, so you can measure the flux that's coming through the pores versus the flux that's going through the channel. Okay, and you do exactly the same measurement that we did before, and you come up with this. It's amazing. It's exactly the same thing. What channel velocity, pore leakage? You get a bunch of different things over here, and they all collapse to something on the scale of 1/2 when you go up to the higher flow rates. Okay, so we think we understand the permeability piece. All right, so that's the permeability piece. Remember, I told you there is a second piece, which is for filtration, which is what size particles escape. Okay, so and that's basically saying, okay, which particles are going to go through my pores and which are going to go down into the stomach of the manta. Okay, and this is a much easier calculation. This is just geometry. All right, so here's the geometric calculation. Again, that's my leaky cylinder. So if I zoom in on one of these little pores, there are a bunch of streamlines that just make it past the tip of this pore. Okay. If I look at this green streamline, so this is a green, so actually let's look at the red streamline. So the red streamline is the one that hits this separatrix right here. So that's the one at which you could go either way. And if I'm above the red streamline, I go down the middle, and if I'm below it, I go into the pore. Okay. If I have a green sized particle on this red streamline, it's fine. It can go past this and it can make this corner. But if I have a bigger particle, like if I try to put this purple particle on this red streamline, it runs into this guy and then bounces back into the flow. So the largest particle that can make it into this pore is the red size of the red. Everybody buys that? Yeah. Okay. So we can check that again because we have an experiment. So here's the experiment. It does exactly that. So this is a time series photograph. So you can see the little guys are making it around the corner, and the big ones that can go around that thing falls off into the middle of the chest. Okay. So we just do that. So this is again just a geometric calculation. So the geometric calculation goes as follows. You say, well, I've got some kind of a parabolic flow in this channel, or other kind of. We'll talk about other shapes as well, but let's start with parabolic flow. This is my critical streamline, right? So this is the largest particle that can sit on that critical streamline. And so anything that's on this side of that streamline is going to go through the pore, and anything that's on this side of the streamline is going to go through the channel. So if I want to know how much leakage I have, I just have to integrate these green regions and normalize that by the integration of the whole parabola, and that gives me the fraction of stuff that's going out the sides. Yeah. I just have to do that calculation. And you do that, and you get a very simple relationship. So this is QL divided by Q channel, right? So that's this green thing divided by the integral of the whole thing over here. And you get some numbers that come out of this. And in particular, it scales like the cutoff particle diameter, which is the diameter of that red guy, squared. Okay. All right. So I'm now just going to plot that. Okay, so I'm going to plot this, and I'm about to tie this all back to mantas. We're almost to the point where I tie this back to mantas. So I'm going to plot this. Okay, so again, this is a log-log plot. So this is the scaling that you get if I put in a viscous profile, which is a laminar profile, which is my parabolic profile. Okay, and again, which is calculated. This is just a number, so it gives you this line. I can also do it for an inviscid profile. An inviscid profile is basically constant across the channel. Right, so it just gives you a different shape. So I can do that as well. So here's my inviscid profile. And the only place in this parameter space that's accessible to a manta or any kind of filter is between these two lines. Right, I can't get more inviscid than an inviscid profile. That's it. That's limit. And I can't get more viscous than a parabolic profile. That's it. Right. So those are so everything that should be between those two lines, which means that all of this region is inaccessible. All right. So this now also gives us a little bit of a challenge if you're building a filter, because what do you want if you're going to build a filter? So if you want to build a filter, you want to be able to filter out very, very tiny particles. That's the whole point of the filter, right? So you want to be somewhere down here. You also want to have a very high flow rate, right? So if I'm building Deer Island, I want to be pumping through as much fluid as I can. So that means I want to be somewhere up here, which means that this is the magic spot on this plot. This is where my perfect filter is. Okay, but I can't be there. So fluid mechanics is already telling us there's a trade-off. You have to pick which one we want in this space. Okay. Again, let's check to see if this is really true. So we do a whole bunch of simulations and see where they sit in this plot, and they all sit there. So yes, we are confined to that space. So okay, good. All checks out. Okay, so now we come back to the manta. So if you are a manta, what do you want? So here's my manta. So what is your goal? What is your goal in life if you are a manta? Food, exactly. If you are a manta, you want to eat. So here's what mantas eat. So this is sort of a pyramid of stuff, and they tend to eat these microplankton over here, which are 20 to 200 microns. So this is the region that's 20 to 200 microns. The other thing, it turns out there's one other thing that mantas need to do, which is that they need to breathe. And the way they breathe is by flushing that fluid through their gills, right? So you need to have a minimum amount of flow rate through the gills in order to get oxygen. Okay, so you need oxygen, and here's the minimum flow rate, which again you can calculate. So they need to be somewhere up here above this sort of green box if you're going to get enough oxygen to live. Okay, so now that tells me with all of these different constraints, my mantas must sit somewhere in here, right? If this is how the mantas are designing those filters, if those are the constraints, they must sit somewhere in there. Okay, so remember, we now have everything we need to solve this problem. Because remember the first half of the talk, which was about permeability, the whole point of permeability was to talk about how do I get my Q leak divided by Q channel? How do I relate that to the geometric parameters of those little plates? Right, so I can go back and forth. I can turn this into a geometric parameter for little plates, and then I can compare that with where mantas actually sit in this plot. Okay, so as far as I know, there are like about 10 species of manta, and so I am now going to put those 10 species of manta on this plot. And if this is what they are doing, if this is the trade-off that they are making, they should all be within that little red square. Okay, so here are the plots. This is actual biological data, so we take the micrographs of this, we measure the distance between each of the plates, we measure the size of the plates, the length of the plates, we measure the swimming speed, and we put all that in the plot. And what you get is this. Amazing! So the mantas sit right in the middle of this little red box. So I would say that this is strong evidence that the geometry of the pores and these mantas has evolved to balance this trade-off between feeding and breathing. Right. So if that's the thing that you're worried about, the trade-off between feeding and breathing, that's the size of the plates that you should make if you're a manta ray. Okay. All right. So that's part one of the talk. I'm going to keep going because I do want to talk about I know there's a lot of people here with a lot of expertise in education, so I do want to talk about the education piece. So I'm going to give you a minute to pause and think about your manta questions, so you have them in your brain for the end of the talk. So everybody, get your manta questions ready, and then we're going to switch gears. All right, everybody's got their manta questions ready in their mind. All right, so part two of this talk. We're now totally going to switch gears. So put the manta aside. We're going to switch gears to open-ended problem solving in engineering education. So let me just go through a few of these slides first. So first, there's been an enormous amount of very good work done in this space, right? And so we did like a lot of literature review to kind of try to understand what people have done in this space. And there are many, many, many great papers. And I wanted to put this up just to highlight this line right here. So people talk about what stage of the problem are you solving, exploring, planning, what types of representations are you using, what's the accuracy of your solution. But what I want to talk about today is the openness of the problem. Okay, so the openness of the problem, there's a whole sort of array of kinds of problems you can assign to students. The ones we typically give them are textbook exercise and textbook problems. We do this all the time in every class, everybody's seen. Okay, you go a little bit further down this chart. You get to problems that have more open-ended solutions. So these are things that start to look more like research, like what we do in a research group when we're trying to solve things where we don't know what the answer is. And there may be many methods that you could use to attack these problems, and there may be many different answers that you could get to these, and some are more right than others, but all of them have some validity in the solution space. Okay, so we wanted to ask, we wanted to think about this space. How good are students that come out of our programs at solving problems that are kind of in this part of the space? Okay. So one of the interesting things about this part of the spectrum, so for all of us that have done research, what we know is that typically speaking, the first method you try is probably not the best, right? You've done this. And it probably can be beneficial if you want to switch to another method. Okay. All right. So now this actually now turns into an interesting optimization problem. So here's the optimization problem. So if you don't switch enough, so if you're too hesitant to switch, you don't explore the solution space sufficiently. Okay. And so you're going to miss some useful problem solving strategies, right? Because you're too focused on whatever it is you're trying to do. On the other hand, if you do too much switching, maybe you have not put enough effort in each attempt, right? You don't dig in deep enough. And so then you abandon your strategy before it pays off, because you do have to do a little bit of work to see if your strategy is going to pay off. So that suggests you don't want to do too little switching, you don't want to do too much switching, which suggests there is a Goldilocks switching, which is an optimal switching propensity to solving problems which have multiple solution paths, right? And I think people, like for faculty who have done this, who have solved problems for many years, many decades, we do this intuitively. We have a sense of when we need to abandon one method and move on to another one. But I'm going to show you that this might not be obvious to students who are doing this for the first time. Okay, so first, I'm actually just going to solve the optimization problem, and then I'm going to show you data from students. Okay, so here's the optimization problem. So I'm first going to do it just doing a Monte Carlo method. So suppose I'm in a situation where, let's suppose it's an exam. So I have a finite amount of time, t final, to solve this problem, right? So I have t, this is how long I have to solve the problem. Okay, and there are many options I have to solving the problem. And I'll give you a bunch of options for estimating the area of something. And I'm going to show you what that problem is later. But for now, you can know if I'm going to estimate the area, one thing is I can just look at it, say, yeah, it's about 4.5. Great. Very short, doesn't take much time, but it's a very rough estimate. There are lots of other things you can do. You could do geometric approximations, you can do CAD, you can do Monte Carlo, you could integrate. Some of these are going to be too long, some of these are going to be almost too long, some are going to be very quick, and you have to select from this array of things. So here's the Monte Carlo that I'm going to do. So suppose the following. Suppose I have this much time, 10 minutes, to solve my problem. And my probability to switch is lambda. Okay, so what I'm going to do is I'm going to randomly pick some starting method over here. Okay, and then on each square, I'm going to flip a coin that's weighted by lambda. And if it says switch, then I will randomly pick a different method. And if no, I'll stay on the current method. Okay, so you'll get something that looks something like this. So I randomly picked integrate, and I flip the coin, and I switch here. So now I have CAD, and then I flip the coin, then I switch to geometric approximation. Okay, and a solution is found if the problem solver stays on one solution method long enough, right? So for example, here I stayed on geometric approximation for 5 minutes. 5 minutes was enough, so check. I solved the problem in this particular instance. Okay. And then I just do this, it's a Monte Carlo. So I do this 1,000 times. 1,000 times, and I just count how many times do I solve the problem and how many times do I not solve the problem. That will give me a probability that I can solve the problem. Okay. All right, so you do that, and you get something like this. Okay, so this is just a particular example. this is with 2 solution methods instead of 1 solution method. And the first one is the faster one. And you can see if I have a very fast solution, so that's this guy over here. If in my box of tricks, one of the solutions is fast, it behooves you to switch a lot until you find your fast solution, right? So this tells you my probability of switching should be something like this, like 0.4 almost on every roll. If my solution is really slow, so my fastest solution takes up almost all the time, then don't switch. You just have to get lucky and hope you pick the right one at the beginning, right? You don't have time to flip back and forth. Okay, so and there's some trade-off in the middle. Okay. The thing that I like about this is there's also an analytics solution to this. So you don't have to do the Monte Carlo. Okay, so I'm going to show you the analytics solution and then I'm going to show you the data. So here's the analytics solution. So the analytics solution, you say, well, okay, I have these sort of random events, which is when I'm switching, which are arriving at random times. So this is, I know there's some physics folks in the audience. So if I say random arrival events, you probably, yes, exactly. It's a Poisson process. So this is a Poisson process. So Poisson process, stochastic process, it models the number of arrivals occurring within a specific time interval that occur randomly and independently. And so for a Poisson process, the probability of B transition, so me switching B times in a time interval TF is given by this. So that's a known relationship. That's the Poisson process. So now the probability of solving the problem is given by this. So here's the probability of solving the problem. This is the problem. So I sum over all possible B's. This is the probability of switching B times, which came from the Poisson process. I have to multiply that by the probability of finding solution given B switches. Okay, so this I know, this I have to find. All right, so let's find this. So I'm going to show you how you find this for a very simple case, and then ask you to extrapolate how you would do this to the more complex cases. All right, so we need to find this. We need to estimate the probability that I solved the problem given that I switched B times. Okay. All right, so consider the following case. So consider that I only have 2 solution methods, method 1 and method 2. I'm going to consider the case where I have 2 transitions, and then I start on method 1. And that method 1 is less than T final, and actually I don't really care what T2 is, but method T2 is larger than method. Okay. All right, so how do I figure out what is the probability given this the 2 transitions that I solved this? So you can just draw a picture, and here's the picture you draw. So this axis is the time of the first switch. This axis is the time of the second switch. Okay. By definition, the second switch has to happen after the first switch. Right. So that part of the diagram's out. Okay. Now, for the first time of the first switch, if I stay on this longer than T1, that means I've solved it. Right? Because I just have to stay on it long enough for T1 and then I got it. So that tells me if I am at T1 or longer, then T1 was sufficiently long to solve the problem. And so if I'm anywhere in this pink region, I've solved it. Alternatively, if I go to T1 and then I switch to T2 and then I switch back to T1, as long as I switch back to T1 with sufficient time that the rest of the time left is longer than T1, I've also solved it. Okay, so that means that this also gives you enough solve, because as long as I switch back to T1 before this bar, then I have enough time up here to solve it. So now the probability that I solve the problem is just the ratio of this little mountain region to the size of the triangle. That's it. So that's the probability that I solve the problem. So that's the answer that goes up here. And if you want to go to 3 solution methods, instead of having this, if you go to 3 switches, instead of having this as a 2-dimensional place, you have it as 3-dimensional, and you have overlapping pyramids. And if you go to 4 dimensions, you have overlapping 4-dimensional pyramids, but they're all just geometric solutions. So as long as you can take the area of those pyramids, you can solve the problem. Okay, so you do that. And you can get an answer. Okay, I'm going to show you the answer in a minute, but I'm going to show you the answer on top of the experimental data. So here's the experimental data. So the experimental data is we gave a bunch of students, it was like 70 some, 10 minutes to solve a problem. Okay, here's the problem that we gave them to solve. So we just said, basically, you have a steel disc and you want to cut this thing out. Please estimate the area of this gray region using whatever method you want. And we didn't tell them why we were having them solve this. We just said, just solve this problem and estimate the area of that gray region. And as they did it, every 2 minutes, we gave them a new colored pen. So we could see the order in which they did things in their solutions and when they were switching, right? So we now have a time history of what they did as they solved the problem. Okay. So what we got out of this is what the students did. So these are all the methods they tried. They did visual estimation, geometric. They did CAD. We say you could use anything they want. So they had their laptops. So some of them actually used CAD, or did Monte Carlo, some integral. This is the fraction correct. Don't do the integral. The integral is super hard on this problem. It's terrible. This is the number of times they switched. So most of the students did not switch. They just picked one and tried to hammer it through. Okay, so 64 of them did that. 7 of them switched once, and 1 of them switched twice. Okay, and you can see that sort of anecdotally, the more you switch, the more likely you are to find a correct solution to the problem. Okay. I'm now going to lay this on top of the analytic solution of what's the optimal number of switches you should make. Oh, sorry, I'll show you this. This is what they actually switched to. So this is like, these are like 27 started on the integral, and a bunch of them tried to keep going with the integral. The color shows how successful they were. So this is light, so not successful. If you switch from the integral to a geometric approximation, you did much better, right? Geometric approximation, they sort of stayed here. So there's a bunch of different ways that they tried different things. Okay. But here's the question we want to know is, are they switching enough? Okay, so here's the question, are they switching enough? So this line here, this is the analytic solution that I just showed you that you do from those summing up all those triangles and pyramids. This data is what we just got from the students, right? So I have one right here, which is this is the students that did not switch. So over here, these are the students that only switched once. And again, I have a few error bars because I only had 7 students that switched, and I only had 1 that switched twice, and then they got it correct. So they're up here somewhere, right? So things seem consistent, and they also suggest, this plot suggests that these guys are not switching up. They should have tried something else, right? But they should have realized at some point, oh, this integral is not going to get me where I need to go in 10 minutes. I should be exploring some other options. Okay. And again, that's consistent with this, where you do better if you switch more. Okay. So the conclusion I want to bring out is that we need more data, right? I only have 7 switchers, so I really need more data. But what we have, the data we have suggests that the students are not switching enough. And why might that be? So one of the reasons we were interested in this problem is that we train people to solve problems over here in these textbook exercises. And the textbook exercises are very linear. It's do this, do this, do this, and then you get the answer, right? So we are not exercising that part of your brain, which is exploring the solution space. To say, maybe I should try something and then get rid of it. And then students are very hesitant to let go of something once they're going down a path, right? And maybe we need to be thinking about how do we make people a little bit more flexible when we're training them over here to get them a little bit more used to trying other options when they get. So anyway, so that's another area that we're thinking about. We're thinking about how do you actually mathematically model where should we be pushing the students to be as they're learning this trajectory. And with that, I'm gonna end. So I'll just say that Jinyu Mao is the student who did all the work on the manta ray. Hopefully you saved your manta ray questions. Told you to take note of them. Irmgard is a faculty member who also worked on the manta ray problem with it, and Wilmy is my student who worked on the engineering education piece. And with that, I'll thank you for your attention.
Thank you so much, Peko. That was absolutely fascinating on both topics. So now I'll open to the floor and see if anyone has any questions. Yeah, Zara.
Hey, thank you for your talk. I thought it was fantastic. So my question for you is, with the flow of going into the manta rays, how robust is it when flow becomes turbulent?
Yeah. So the calculations I showed were all for laminar, but we have also done it for the turbulent case, where if we have channel flow and we have a turbulent flow profile, since you for within different kinds of limits, you can write down a turbulent flow profile. And everything that we've said goes through. And in fact, when we actually looked to compare the actual manta data with the points on there, we used a turbulent flow profile because the manta is going at a Reynolds number, which is low Reynolds number is not going to do it. So that was a turbulent flow profile when we did the comparison with the animal.
Just follow up on that. So your numerical simulation for the laminar flow indicated that you had a single big vortex, but in a turbulent case, would there be some distribution of vortices?
Yeah, great question. So we have put a turbulent flow profile in there, and we still get a single vortex. Oh, I don't know what I just did. Continue to share. Because I'll show you if I show you right here. So certainly there probably are regimes where you get more than one vortex. Oh, sure. So the turbulent flow is up here where I have a high Reynolds number. And this gap is always still pretty small, right? So in this gap, you're sort of limited in what the flow can do. So as long as this is sort of millimetric or smaller and we're using water, you tend to get just one. Yeah.
Oh, sorry, there's one
Oh,
just here,
I'm
and then
Sorry.
one there, and then one.
Okay, I'll
Sorry, Rani.
Sorry. Great talk. I just wanted to ask about the application of this work to a sort of industrial scale filtration plant. Your sort of model looked pretty small. Do you have plans to scale it up? And do you have some idea of how much more efficient this is than the current method?
Oh, great question. Yeah, great question. So I haven't done the efficiency calculation to compare what your island is currently doing. I think one thing to think about is so when you're thinking about these filtration plants, there are all different levels of sizes of particles and different kinds of things to take out of the flow. So one of the things that we need to think about is at which stage is this more efficient than what they've got, right? Or even if it's not more efficient, is this as efficient but it won't clog, right? So I think it's the lack of clogging that makes this really attractive because you're filtering out particles that are smaller than that gas size. So I think that can win at. I think the pumping should be similar, right? Because again, you're sort of we've looked at kind of the parameter regimes of where you might be pumping. The question is sort of how small can this go, right? So here we're filtering out particles that are maybe, I don't know, like I think I said 20 to 100 microns. Whereas in Deer Island, you do have to take things out that are smaller than that. So you might want to do this as a first stage and then put it through a minor filter.
Thank you for very nice talk. I have a question about the educational part. In your problem classification on the right, there were wicked problems, or maybe they can be called problem straps. Do they offer them, and could you comment anything about them?
There's something that's called wicked problems. So if we go over here, and the way we think about this, although again, people have thought about this in the engineering literature in many different ways. So over here, chair. So at the very end here, we have something that's called wicked problems. We did not look at those in the study. One thing that I would say about this is that these problems are probably hard to do in a fixed time scale. Like, so you wouldn't want to give somebody this in 10 minutes. So one of the things we did do is we looked at the dual problem to what we looked at. So the problem I showed you was if I give you a fixed time period, what is the probability that you solve the problem? The dual problem is to say, I give you an infinite amount of time. What is the switching strategy that minimizes the amount of time you would need to solve the problem? Right. So that's more of like a PhD problem, right? And so that I think that the dual problem is probably more relevant to things on this side of the spectrum. But I also think that these are complicated enough that there are other traps you could fall into. So I want to think about it a little more carefully.
Thank you.
Yeah.
Yeah. Can I take you back to the slide you had on the manta ray?
Yeah. Which one?
Just the one that you had with the vortex.
With the vortex. Yeah. Yeah. Right here.
So you said the biologists were explaining this as a hopping solution where the particle hops from one obstacle to the next. And I was wondering, with the vortex there, would the particle kind of flow on top of that streamline and have a much narrower channel to get into and actually hop out again if you...
Yeah. So, oh, that's a really interesting suggestion, right? So you're saying that my particle, let's say I have a bigger particle, and for some reason it gets caught in here. If it's big enough, it's actually not going to get into this part of the flow. It's going to get caught in the vortex, which means it's going to come around and then pop back out of the vortex. So we have not looked at what happens when particles cross streamlines, which would certainly be true because there is some diffusion in these problems, and this is a high enough Reynolds number that there's probably some inertial effect that will allow them to cross the streamlines. I had not actually thought about this trajectory, so that's a good one. Well, we'll have to take a look at the experiments and see if we see it.
So we'll take one more question from in the room and then we'll open to the people on Zoom.
If there's any rotation in the flow, is that going to segregate your particle sizes between the wall and the interior of the flow?
Yeah, great question. If I've got, let me just put my channel. Yeah, so one of the things, so we have sort of assumed that the particles are uniformly distributed. And so the question is, that's not necessarily the case, right? So there are certainly cases in channel where owing to shear effects, owing to inertial segregation, owing to rotational effects, where particles can be sorted by size or can congregate at different parts of the channel. I think about this. So the answer is I have checked. And I have to look to see. So I will say that in the channel, the flow in the actual manta tends to be pretty well mixed. But I hadn't thought about the rotational segregation. I'm pretty confident there's not going to be shear-induced segregation, and I think inertial lift is not going to do anything, but I'm less confident about your rotational.